diseases or infection. And not only that, even you can observe the garlic. So I think most of you know about garlic. So nowadays, during cooking in our homes, we are using the garlic frequently. So if you are using garlic, automatically the garlic, which is also having so many health benefits. So garlic, which is one of the component which is belongs to the bulbs of Allium sativum, belongs to the family of Liliaceae. So which is having ability to reduce the cholesterol. So fat and lipid levels that are going to be reduced with the use of garlic. And it is also having ability to detox the harmful toxins present in our body. And it is also having the ability of immunity booster property. And it also reduce the body weight. And also involved in controlling hypertension. So for the treatment of hypertension, the garlic, which is also very useful, so that when we are doing cooking at our homes, we can use this garlic, which is having a lot of benefits. So not only that, every day, daily morning, if you are going to take just 2 bulbs of this garlic in empty stomach, it will give a very huge health benefit to our body in reducing cholesterol and our detoxification process. And coming to the garlic related medicine that is available in the market currently, garlic pearls. So many people nowadays, they are using these garlic pearls, which are having ability to treat various heart diseases like hypertension. It can reduce cholesterol levels, and it improves the digestion process of our body. So which is the very helpful to our body. Similarly, even the garlic oil, it is also available in the market. The garlic oil, it promotes the hair growth and also improves the skin texture and soothes the rashes and providing rash free skin. So it completely soothes the rashes. Not only that, even it can improve our hair growth. So garlic oil is available, garlic pearls are available for the treatment of various diseases. And coming to another one, that is tulsi. It also comes under the Ayurvedic one. So tulsi, which is obtained from Ocimum sanctum, belongs to the family Lamiaceae. It is having so many health benefits. It includes anti-inflammatory activity for the treatment of inflammation, and which is blood purifier. It purifies the blood and removes the toxins in that blood, and it acts as antipyretic agent. That means it can reduce the elevated body temperature and having antifungal property for the treatment of fungal infections, anti-diabetic property for the treatment of diabetes, anti-stress, it can reduce the anxiety and stress, antibacterial property, and it is also having antioxidant property, analgesic property, and it is also immunomodulator in nature, having carminative property, cardiac toner property, beautifying property, and it also improves the digestion, and anti-aging property, anti-allergic in nature, and anti-cancer property and stimulant. So you will see which is having a huge health benefits. And what are the various types of products that are available with this tulsi in the market? So here you can see Baidyanath Giloy Tulsi juice. So this Baidyanath Giloy Tulsi juice, which is nothing but Tulsi Panchang juice, which is used to relieve the cough, and it also improves or empowers the body immune system. And another one that is produced by or manufactured by Himalaya, that is Pure Herbs Tulsi. So this Pure Herbs Tulsi, which is manufactured by Himalaya, it can be helpful in case of various respiratory problems, and it also improves the immunity, and it also having antimicrobial property, and it fights against the respiratory infections. So such type of health benefits are there. So these are the some of the formulations that are available in the market with this tulsi. And similarly, coming to ashwagandha. So ashwagandha, which is also called as Indian ginseng, botanical name Withania somnifera, belongs to the Solanaceae family. And coming to the health benefits of this ashwagandha, it improves the fertility and vitality, and it also stimulates secretion of thyroid hormone T3 and T4, thyroxine and tetrahydrothyronine, T3 and T4 hormones, which are going to be stimulated. Secretions are going to be stimulated with this ashwagandha, and it is also effective in controlling the bacterial infections as an antibacterial agent. Also boosts our body immunity system and reduce the seizures and convulsions. Also relieves stress, anxiety, and depression. By reducing the dopamine levels in our body, it can reduce the stress, anxiety, and depression. And it also provides in relieving from inflammation and pain, and having beneficial effect in treating the diabetes and cataract. Diabetes and cataract for the treatment of eye-related disease, cataract. And it also strengthens heart muscles and controls the cholesterol. That means it can reduce the body cholesterol levels. So these are the huge benefits of this particular ashwagandha. And coming to the available products in the market related to this ashwagandha. So Himalaya is manufacturing organic ashwagandha formulation, and even ashwagandha capsules which are also available. So use it for the reducing the stress and anxiety and depression conditions, and it also improves our body immunity system. So these are the various Ayurvedic herbal medicine that are generally used since from our traditional system. From olden days, ancient days itself, we are using these products. And coming to the Siddha medicine. So Siddha medicine, it is also an ancient Indian healing system. 10,000 years old traditional system, which is introduced by in Tamil system of medicine. Siddha, it is a Tamil word derived from Siddhi. And coming to the Siddha medicine, this particular traditional system of healing that originated in, especially in South India portion of our country, and is considered to be one of the India's oldest system of medicine as like Ayurveda. And it is based on combination of ancient medicinal practices in combination with spiritual disciplines as well as mysticism, that is nothing but belief. So with all these combinations, this is called as Siddha. And some metals which are used by Siddha system like arsenic, lead, mercury, copper, silver, and are deadly toxic. I think most of you know that all these metals which are having some toxicity. But in fact, in ancient days, the Siddha system recipe, when they are going to perform some purification process and mixing with some other materials, Ayurvedic materials, that leads to reduce the toxicity of those metals after making the recipe of that Siddha medicine system. And it reduces the toxicity of those metals used in that Siddha system. And these preparations are really effective. And that particular system or recipe that only can be explained by only Siddha experts only. And from the scientific point of view, such possibility is currently it is in remote only. The scientific evidences are very less. And coming to some of the preparations available in the market under this Siddha, TKN Siddha Ayurveda Vaidyashala, they are preparing Vallari Churnam. So this Vallari Churnam, which is the Siddha preparation, which is having neuroprotective nature, and it also helps in relieving the or reducing the wound healing process, involving wound healing process, and relieves anxiety and stress conditions, and it improves memory power. That is nothing but cognition. And it also acts as blood purifier and fights against various skin conditions, and it can be remove toxins from our body. Those are the advantages with this Vallari Churnam. And another type of Siddha preparation, which is produced by Zendu. I think most of you know that Zendu, leading manufacturing company. So Zendu Siddha Makardwas Gutika Gold tablets. So the advantage of this particular Siddha preparation, that is, it improves the strength of the heart muscles especially, and also it acts as anti-aging agent and acts as antipyretic and dyspepsia, in reducing dyspepsia, and helps in improving the immunity system. So those are the advantages with this Zendu Siddha Makardwas Gutika Gold tablets. Then coming to the Unani medicine system, this is also one of the oldest system of medicine as that of Ayurveda and Siddha, and it is especially particularly this Unani medicine system which is originated in Greece. The theoretical framework of Unani medicine is based on the teachings of Hippocrates. who is known as the father of the system of medicine. And he is involved in introducing so many of medicine, superstition and magic in 1460 BC. And according to this, Hippocrates theory poses the balance of elements, humors and temperaments that keeps the body and mind healthy. So by introducing these things, the Unani medicine system was developed in Greece. In continuation, the other Greek scholars followed the Hippocrates footsteps and they contributed a lot in the development of this Unani medicine system. The later, the Arabs developed and refined Unani medicine system through the systematic experimentation processes, particularly by the scholar physician Avicenna in 980 CE. And this Unani medicine system also received contributions from other parts of the world, that is from Middle East and South Asia regions, and came to be known as Arabian or Islamic medicine system at one point of time. And these Unani medicine practitioners in generally use a vast materials of medica derived from natural sources, including herbs, minerals, and various components of animal parts, like that. The Unani pharmacopoeia consists of more than 2000 recipes of the medicines and medicinal preparations, which can be in the form of powders or decoctions or infusions, oils, ointments or creams, etc. And here you can see one of the formulation belongs to this Unani medicine for cardiac disease. So it relieves the joint pains, muscle pains, and cardiac diseases also that can be reduced, and even it can reduce the body pains, body ache. And another preparation available under Unani system, Hamdard Majun Hajrul Yuhat powder. So which is also available currently in the market. It is used for the treatment of uterine infections and also for the kidney stones, reducing the kidney stones and to remove the small kidney stones from the gallbladder, and urethral and bladder stones that can be removed by using this particular powder. And coming to naturopathy. So after Ayurveda, Siddha, Unani, we are having naturopathy. So I think most of you know that naturopathy, it is also known as naturopathic medicine. It is a type of holistic approach to improve our health system that uses natural remedies and therapies to help the body healing system. And naturopaths are practitioners use variety of non-invasive practices that are often branded as natural or self-healing process, like that. Why? Because they are using some natural remedies, so that they are mentioned or branded it as natural or self-healing medicine, like that. The goal of this naturopathy is mainly to promote wellness by supporting the body's natural healing process rather than simplifying the treatment of symptoms. So that means it can improve our body healing process rather than treating the symptoms. And coming to the main aims of naturopathy. So the main aims of naturopathy, it includes educating the people to look after their own health and also minimize the symptoms of illness and balance the body so that illness is less likely to occur in future. So if we can adopt the naturopathy techniques, so in future we are trying to less likely to occur in future related to various diseases or infections, and address the root of cause of a particular person's illness. So this is the main aim of naturopathy system. And earlier there was no compact medical system in India. So because of no compact medical system in India in olden days, because of that reason, people they are using Ayurvedic system or Siddha system or Unani or naturopathy, like that. Even not only that, yoga currently, that is also one of the system. But in olden days, most of the cases they are using Ayurveda, using various herbs for the treatment of diseases and infections. Gradually, the people began extensive research work in the area of medical sciences that leads to discovery of huge number of molecules which are produced for the treatment of various diseases and ailments. And different research scholars and people as well as the rulers contributed immensely towards the development of medicine in the worldwide. So because of that reason, nowadays the complete scenario that is converted from traditional medicine to the novel drug molecules. So even though nowadays there are so many people in the worldwide, they are using ancient traditional medicines, Ayurveda, homeopathy, some people they are using Siddha, some people they are using Chornas like that. So which are also having some good health benefits. So coming to the traditional medicines to novel drug discovery approaches. So with that traditional medicinal system, nowadays the novel drug discovery system was introduced, and that novel drug discovery system, it includes the development of new chemical entities through various techniques, that is designing of molecules. After designing of molecules, identifying the lead molecule through various techniques, that is combinatorial chemistry, high throughput screening process, QSAR studies, then followed by primary assay methods, that is nothing but high throughput screening and in vitro methods, secondary assay methods, in vivo methods for identification of toxicity, metabolism, etc., absorption, distribution, metabolism, clinical studies. With all these techniques, the novel drug discovery process that is involved. And before that, just I would like to explain the novel drug discovery, how it is going to be happened in ancient days with the traditional drug medicines. If you can see the traditional drug medicines, for example, I think most of you know that the cinchona bark, which is nothing but one of the alkaloid consisting of quinine and quinidine. So which is also called as quinine bark. Botanical name, that is Cinchona officinalis, belongs to the Rubiaceae family. And the cinchona bark consisting of mainly the important chemical constituents are quinine and quinidine. If you can see the structure of quinine and quinidine, so you can observe the structure of quinine and quinidine. Both the structures are, by looking, both the structures are resembling same, but there is a small difference in their stereochemistry with respect to a particular point. Because of that particular difference in the stereochemistry point, the quinine, it behaves as anti-malarial drug, and quinidine, it behaves as anti-arrhythmic drug. So here you can see the difference between the quinine and quinidine. Both are structurally similar. The number of carbon atoms, oxygens, nitrogens, hydrogens, same. Molecular weight is also same, but the difference is their stereochemistry with respect to this particular 8th and 9th position. So in case of quinine, the stereochemistry of 8th and 9th positions, that is 8s and 9r configuration. But when coming to the quinidine, 8th position R configuration and 9th position S configuration. Because of that stereochemical difference in the 8th and 9th position, one of the molecule it behaves as a anti-malarial property, another molecule it possesses anti-arrhythmic property. And the novel synthetic anti-malarial drugs which are developed by taking this quinine. So here you can observe the structure of quinine, it consisting of quinoline moiety, and 4th position we are having that attachment, quiniquidine system, that is present at 4th position. So through various QSAR technique studies, the 4th position of this particular quinine, which is going to be replaced with some amino alkyl amino side chain. So with that chain, the synthetic antimalarial drugs which are introduced into the market. Can anybody, you can mention what are the synthetic antimalarial drugs? Any of the participants? Can you name the synthetic antimalarial drugs? The synthetic antimalarial drugs, it includes 4-aminoquinolines, 8-aminoquinolines, which are also having same quinoline moiety. Here you can observe the chloroquine. The 4th position of that quinine, which is replaced with some new alkyl chain, which are connected to the nitrogens, that is amino alkyl amine moiety that is introduced at 4th position. So that chloroquine molecule that is introduced into the market. Not only chloroquine, in that category, there are so many drugs which are introduced into the market, that is chloroquine, amodiaquine, chloroquine, derivative, hydroxychloroquine, piperaquine, like that. All those are 4-aminoquinolines. Further, 8 amino quinolines which are also developed from the traditional quinine by changing the structural moiety, 8th position. The same amino alkyl amino side chain which is introduced, now it is at 8th position. By introducing that moiety at 8th position, 8 amino quinolines are introduced into the market. That is pamaquine, primaquine, tefenoquine, like that. So nowadays, the quinine traditional medicine which is replaced with this chloroquine, hydroxychloroquine, like that. Even during COVID time, so many people they are using this hydroxychloroquine. Even nowadays, chloroquine which is widely used for the treatment of malaria. So like that, the traditional medicine system which is transformed into the new synthetic molecules development process. And not only this one, the drug discovery process which is involved in through the various in silico processes, that includes computational approaches. Computational approaches which are involved starting from the development of chemical library, followed by identification of hit molecules through the high throughput screening process. Then from the hit molecules to identification of lead molecules through the process of lead optimization, followed by preclinical trials and clinical trials to identify the pharmacokinetic parameters and ADME parameter, that is nothing but pharmacokinetic parameters, absorption, distribution, metabolism, and excretion, and to identify the safety, dosage, efficacy, and adverse effects of the molecules. Now, just I would like to explain some brief information related to this particular computational approaches which are involved in drug discovery process. Coming to the computational chemistry, combinatorial chemistry. So, under combinatorial chemistry is nothing but it is a technique, systematic and repetitive process by which number of molecules that can be developed within a short span of time, even with less manpower. That is the advantage with this combinatorial chemistry. I think most of you know that in olden days. So for the synthesis of one molecule, we are going to utilize reflex condenser process, like that. So by using one reflex condenser setup, we are going to produce only one molecule. So with that setup only we can produce one molecule. If we are going to produce 10 molecules, we have to set up 10 reflex condensers. But by using this combinatorial chemistry, the instruments which are available for this combinatorial chemistry itself, which is having huge number of wells, the same temperature and pressure conditions that can be applied to each and every well of the instrument, so that we can produce the more number of derivatives of that particular pharmacophore or ligand molecule that can be produced within short span of time and with less manpower. That is the advantage with this combinatorial chemistry. So here you can see, this is one of the type of combinatorial chemistry instrument. You can see different number of wells which are present in the same instrument. So we are going to produce, for example, 10 derivatives of that particular pharmacophore. So we are going to use different building blocks of A and different building blocks of B. For example, here I would like to prepare 9 derivatives. So I would like to take A building blocks 3 and B building blocks 3, so that each building block of A is going to be reacted with each building block of B, so that we are going to get 9 different compounds. If you are going to take 4 building blocks of A and 4 building blocks of B, we can get 16 compounds. If you can take 5 building blocks in A and 5 building blocks in B, we will get 25 compounds. So like that, we will get a large number of molecules that can be produced within a short span of time by using this particular combinatorial chemistry technique. That is the advantage and current advanced technique for the synthesis of huge number of molecules within a short span of time. And after preparing huge number of molecules, from that we are going to identify the hit molecules. So hit molecules can be identified by using high throughput screening process. The use of high throughput screening process, it is the automated equipment to rapidly test the thousands to millions of samples for biological activity at the model organism levels and cellular pathways or molecular level against an isolated proteins using in vitro methods. And the instruments used for high throughput screening process consisting of different densities of plates ranging from 96, 192, 384, 768, 1536, like that. All these numbers are just multiples of 96. So such type of wells that are available in that high throughput screening equipment. And after identifying the hit molecules. For example, initially by using combinatorial chemistry technique, for example, we are preparing 1000 molecules. After preparing 1000 molecules, we are going to find out the hit molecules by high throughput screening process. So hardly, for example, we can get 20 molecules are identified as hit molecules. So from the 20 molecules, we are going to identify the lead molecules. So to identify the lead molecules, we are going to perform lead optimization process. It includes QSAR studies, molecular docking studies, ADMET studies, etc. So coming to ADMET studies, so there are different ADMET prediction tools which are available. So here I had given the various tools that are available for prediction of ADMET properties. So most widely used tools for the ADMET prediction process, that is here you can observe Swiss ADME and PKCSM, which are freely available tools in the Google. So we can easily perform the prediction of ADMET parameters of that particular molecules. So you can go through that Swiss ADME engine, which is available in the Google. So you can click on this window. Automatically you can be directed into the Swiss ADME tool. So there you have to draw the structure of your molecule. So here you can see that I will show. So if you can enter into the Swiss ADME software, you will be directed to this window. So here itself, just you can draw the molecule structure on the left hand side provided place. So after drawing the particular ligand molecule or Farnsworth structure, just you can click on the middle part button, red color button, the structure which is converted into the Smiles format. So after converting the structure into the Smiles format, just you can click on the run button. If you can click on that run button, it can generate the ADMET parameters of the particular molecule like this. So after generating this, here you can observe the physicochemical properties, lipophilicity properties, water solubility properties, pharmacokinetic properties, that is a GI absorption, blood-brain barrier permeation property, drug likeliness, that includes Lipinski rule of violations, all those things that can be predicted for that molecule. Even you can get download of all those values which are predicted, so that we will get some prediction values of the particular molecules. Then after that, another important tool, that is molecular docking studies. It is also one of the computational method. Molecular docking studies is nothing but, it is the technique, computational technique, that is nothing but in silico method, which is used to find out the interaction between the two molecules. So here interaction between the two molecules means, let us consider drug molecule or designed molecule, that is nothing but considered as ligand. So generally the drug molecule, it is going to be bind with the receptor or target site. So let us consider the target site, which is nothing but it is protein or enzymatic in nature like that. So drug molecule is going to be bind with that receptor. So drug and receptor interaction, that is going to be predicted by using these molecular docking studies. So we are going to find out whether the particular designed drug molecule is having good binding affinity at the target site or not. That can be identified by using molecular docking studies. So the process of molecular docking studies, it involves. So for the molecular docking studies, we need mainly two components. One is ligand molecule, that is nothing but designed molecule, and another one is target site. So we are going to find out the interaction of this particular ligand and that particular target site. So for the docking process, initially we have to select these two components and we have to make them ready for the docking process. So we have to follow the Lipinski rule process, and even for the after selection of target, we have to find out the if there is an extra water molecules are there, you can remove extra polar hydrogens if it is required, you have to add like that. You have to make it prepare for the docking process. So after preparing the ligand and target for the docking process, then the final step is docking. So with the docking, we can get the docking energy value. With that energy value, we can say that whether that particular ligand is having good affinity at the binding site or not, in comparison with the standard ligand. So like that, we can do the docking process. So here you can see the some of the target sites that can be downloaded. So here you can see various sites are available. For the DNA sequences, we can use DDBJ, EMBL. For protein sequences, we can use PDB. data bank. So most of the cases nowadays,
ఓపెన్ చేసి పెడమన్నాడు
purpose, we are going to use that PDB
పైన హెడ్డింగ్ ఏమో ల్యాబ్ అని
database.
ఉంది.
So with that PDB database, we can download so many protein targets and ligand molecules. So even the ligand molecules which are also obtained from the PubChem, ACD, Zinc database and ligand database and drug bank and ChemDB like that. So already existed drug molecules which are available from these databases, not the newly designed drug molecules. In case of newly designed drug molecules, we have to prepare them for docking process on our own basis. That is required. But newly designed molecules which are not available in these data banks. And while selecting the ligand molecule, we have to obey the Lipinski rules. So the Lipinski rules indicates Lipinski rule of 5. That means the ligand molecule molecular weight should not be not more than 500 Daltons, and the number of hydrogen bonds not more than 5, number of hydrogen bond donors, number of hydrogen bond acceptors not more than 10, and even the partition coefficient value, that is nothing but log p value, that is not more than 5 for octanol and water system. It has to obey for this Lipinski rule of 5, which is important for the activity and selectivity at the target site, as well as drug likeliness properties. And here I have mentioned some softwares available for the molecular docking process. The free softwares are Autodock and UCSF Dock. These are the free softwares which are available in the market. But coming to the commercial softwares, Schrodinger, which is also called as Glide, Gold Dock, Flex Dock, MolSoft Dock, Surflex Dock, all these are commercial softwares. So if you want to learn about the Autodock process, so there are so many tutorial videos which are available in the YouTube. If you can keep on practice on that, just within 1 week, you will get the complete idea of Autodock process, the free software. So here I have mentioned some 2D and 3D structures of the particular designed ligand molecule at a topoisomerase 2 target. So here one of the ligand molecule consisting of some indole moiety Schiff's base compound, its binding affinity that was tested at the topoisomerase 2 target protein. So the topoisomerase 2 target protein PDB, which is obtained from the protein data bank, that is nothing but 3QX3. So at that target, this particular molecule, which is docked, and we got the good results, good energy, binding, affinity that is going to be observed with the standard molecule. So these are the 3D and 2D images of that particular designed molecule at the topoisomerase 2 protein target. At the same time, we are going to find out the docking affinity of the standard molecule. So topoisomerase 2 inhibitor is nothing but generally anti-cancer property. Doxorubicin, daunorubicin, all those are nothing but anti-cancer antibiotic compounds. For that purpose, we are selecting the one standard molecule, that is doxorubicin. Doxorubicin binding affinity at the topoisomerase 2 protein. So like that, we have to do the molecular docking studies. And finally, coming to the conclusion. So in this session today, we have discussed on various traditional medicines. So that is related to Ayurveda, Siddha, Unani, some naturopathy, and not only that, even homeopathy system is also there. I think most of you know. Even currently, so many people they are following homeopathic system also. And these traditional medicines to novel drug discovery approaches that we are discussing, and followed by computational drug designs, how it is involved in development of new drug molecules with the use of in silico tools like combinatorial chemistry method, high throughput screening process, molecular drug study techniques that are involved in drug discovery process, and even ADMED prediction with the use of Swiss ADME and PKCSM tools. So with this, I would like to conclude the session. If you have any queries.
Thank you, sir. Thank you for your valuable speech, sir. The topic you have covered, that is very innovative and very informative. Now, RVS Maunika, Assistant Professor, Pharmaceutical Analysis Department, she will proceed with the vote of thanks.
Good morning, one and all. R.S. Monica, Assistant Professor, of Pharmacy. It's my privilege to deliver the vote of thanks today first on behalf of this collaboration with which my heartfelt gratitude to all those who made this event success. I also extend my sincere thanks to our esteemed speaker, Dr. K. Srikant Kumar sir, for sharing his knowledge and expertise in the field of traditional to research in drug design and development process. Your sessions are regarding the herbal products or Siddha, Ayurveda, and naturopathy, which is leading to the process of synthesis from these natural products to the synthetic drugs, and also the process of confirming the characteristics of the drugs, and regarding the combinatorial chemistry, hits identification, and some of the software for the docking system.
And just finally, I would like to suggest a few points as a pharmacy person. I would like to suggest a few points to improve our health system. So just you can observe this image.
Yeah, that also, sir.
Mostly nowadays we are going to purchase all the provisions from the market in packed manner. So my suggestion is don't go for purchasing of such packed food items. If you see in our olden system, in our traditional system, so all these items which are prepared by our own at our homes itself, purchasing turmeric rhizomes, then we are grinding ourselves at home, so that we are using natural components. But nowadays, most of us we are using to take all these packets which are purchased from DMart like that, some Reliance malls like that, which consisting of so much of adulteration. So as a pharmacy, people try to avoid such type of things. So even for
informative topic, sir.
Once in a month or twice in a month, you can make it prepare in your own at your home. Even not only that, even the fruits which are available in the market, mostly the ripened fruits which are available in the market by using some chemicals. So don't go for that. Just you can purchase the unripened fruits and make them naturally, allow them to ripen in your home. And you can use the natural herbs which are available in our villages and in our area. Don't go for packed foods. Don't use the such type of packets. Garlic paste packets nowadays which are available in the market. Don't use such type of packets. In your own, you can spend some time. It is our health. So we have to take care of our health. Not only our health, our parents, our children health, we have to take care. So my sincere request to all the participants, try to use the natural herbs and we can prepare ourselves at our home for our
That is a thinkable thing. So I think everyone is following now today. I think most of the people have a thought regarding these natural things. Using
Yes.
and preparing at home is a very nice thing. I think most of the people are now following that too.
Yes.
And thank you for being with us, sir, and thank you for your giving your informative session with us. I would also like to thank all the participants for your active involvement and enthusiasm throughout the program. Your eagerness to learn and engage in discussion has made this first day FTP a truly interactive and enriching experience. And a special thanks to the organizing committee, technical team, support staff, and everyone who worked behind the scenes to ensure the seamless execution of this first day FTP. And I would like to finally thanks our principal, sir, Dr. Kishore Babu, sir, King's College of Pharmacy, and the team members of the IPGA and Apredi, sir. And thank you everyone for this wonderful session. Thank you, sir.
Thank you, madam. Thank you for giving this opportunity, especially to the principal of this College of Pharmacy, Dr. M. Kishore Babu, sir, for providing this opportunity, and President of IPGA, Vijay Bhaskar Reddy, sir, and Dr. P. Srinivas Babu, sir, General Secretary of IPGA State Branch, and all the organizing committee members of this College of Pharmacy, Dr. P. Swati, madam, Devadas, sir,
Thank
and
you.
Monica, madam. Thank you all for giving
Thank you,
this
sir.
opportunity.
Thank you so much, sir. Thank you for your valuable speech, sir.
Thank you, madam.
Thanks. Any queries?
Any participants, if you have queries?
Sir, can you tell regarding smiles, the structures, it converted into smiles now.
It is very simple, madam. Just you can enter. Just I will show. Here you can see the Swiss ADME tool is there now. So in
Yes.
Google, just you can enter Swiss ADME like that, so that you will be directed into this window. So this is the link, Swiss ADME link, http://swissadme.ch. If you can
Okay,
click on that,
free tool, free
if you
tool.
if you can click on that, you will be directed to this window, madam. So
Okay.
here on the left hand side, there is a space to draw the structure. So
Okay.
once you can draw the structure as like ChemDraw, then after that, just simply you can click on the middle button, middle arrow, which is converted
Yeah.
into smiles notation. Why? Because
Yes.
the particular ADME parameters which are going to be produced with respect to through the smiles notation. So initially we are giving the molecule in the form of structure. But
Yes,
as
yes.
per its request, as per the software, which is initially converted into smiles notation. Then further
Okay.
that smiles notation is going to be utilized to produce the prediction values of ADMET parameters.
Yes, sir, yes. So
Like
IUPAC name also will get now, sir, by using this software.
no, madam. IUPAC name we won't get.
Okay.
For IUPAC name, we have to use ChemDraw or ISI square like that.
Okay. Only
Yes, yes.
ADME reports we'll get.
Only
by
ADME
using-
parameters like this we will get. Physicochemical properties, that is molecular weight, molecular formula, number of heavy atoms, number of aromatic atoms, number of rotatable bonds, lipophilicity characters, log P values, water solubility property, then pharmacokinetic properties, that is absorption, GA absorption, blood-brain barrier permeation property like that, Lipinski rule of violations, drug likeliness. These things we will get.
Yes, sir. Very nice, sir. Traditional medicine. This was a very innovative and very informative topic, sir. Okay,
Thank you,
sir.
ma'am.
Today we'll end this session now.
Okay, ma'am. Thank you.
Okay, sir. Thank you.